Sure. My background is somewhat tortuous. Took a number of diversions. I started out wanting to become a physicist, actually, and tried that for a few years until one of our faculty members at Penn, where I was doing graduate physics, walked into a lecture and said that National Science Foundation funding decreases since 1969 for the first time since Sputnik, and that most of you won't be getting jobs. That caused a reevaluation of our career paths for many of us, and about half the class deserted that year. I hung out for about another year until sort of reality set in. And I was teaching undergraduates at Penn because I was a grad student. I wasn't really much older than them. Quite a few were pre-med, and so I started asking them, what it was like, what they were thinking about, what they wanted to do, and why, and decided that it sounded like a good thing for me, good fit. Medicine wasn't going to close any doors for me. It was just going to open them because, I could do medicine and, I could become a doctor, but you could also be a hospital administrator. You could go into research. There really were a lot of different things that you could do. Okay, that's a nice avenue for me. And that's what I did. And went to medicine, applied to medical school, got in, and I became an internist, actually, originally. I did that for a few years and found, however, that actually in my residency, there were two role models, and I couldn't see doing either one of them long term. One of them, the docs were leaving at 3 in the afternoon and not taking care of their patients, and the others were staying until 11 o'clock at night and living in the hospital. I was thinking that's not going to be a life for me either, because that's the one I'm going to end up being. So I liked the person doing nuclear medicine at my hospital, which was Kings County Hospital at the time in Brooklyn. And he was a gentleman and a scholar and a teacher, and I liked it, and I liked the field, because nuclear medicine kind of had a lot of what I liked about internal medicine, the puzzle, the diagnostic process, the physiology of what was going wrong. And so I switched to a nuclear medicine program and got into nuclear medicine. And that was around 1980 when I finished my training. And yeah, what are we up to? I'm not sure. Yes. Anyway, I did nuclear medicine and did that very happily for more than 20 years. And then in the 1970s, Richard Feynman's been a friend and colleague and collaborator of mine for many years, and most of the time we've basically BS'd about how we were going to do some research together. And then finally, in the early 2000s, I started to come to his lab, and we started off doing some atomic force microscopy, which didn't pan out. And Richard always had an interest in carb-restricted diets. He'd had an on and off weight problem and had basically avoided the Atkins diet, or he stayed on it briefly but would go off because he thought it was dangerous. And then one day he said to himself, I'm a biochemist, maybe I ought to look at this more closely, and he got interested in it. And our division sort of parted very naturally, because I was interested in the Warburg effect since medical school, and in fact the Warburg effect plays exactly into what I did in nuclear medicine. And PET scans show explicitly that most cancers have glucose uptake, because the radiotracer that's used is fluorodeoxyglucose, so it's quite apparent that that's what's involved. So our division became quite natural in low-carb diets. He sort of went after obesity and lipid metabolism and diabetes, and I was interested in cancer. So that was around 2003. Ketogenic diets and cancer, and in particular, we did one clinical trial, which was a small pilot study of 10 patients. And I've spoken about that before. In previous times, we published a paper in 2012 on this, and the work got started in earnest in 2006, and it took 4 years to recruit 10 patients. It wasn't easy. But around the same time, we had started cell culture work. And I've actually never spoken about that publicly before. I've given a poster on it. published it, but I've never actually talked about it. So I wanted to assemble a talk which described the cell culture work as well as the human trial, because the cell culture work speaks a little bit to mechanism, and the human trial is, interesting for other reasons. I've kind of laid out what I want to do over the next few years, and the things that I'd like to do are going to be hard enough and to be able to accomplish them. So right now I'm very eagerly anticipating going on to do a study that I hope that Epigenics will will support. I mean, we've been in discussions with this, and that's a trial which we're looking to do of 65 patients, 45 of whom will be on a ketogenic diet, and 20 will be on a control diet, which was absent from the prior study too. So we'll be addressing two deficiencies of the prior study. One was that it was very small, and second, that it didn't have controls. And that's going to take me a few years to get done, so I think that's enough for now. But no, the other thing, actually, I should mention Stephanie Mattingly, because she graduated from University of Kentucky, Louisville as a PhD chemist about a year ago, and got in touch with Richard and myself wanting to develop a postdoc fellowship for herself in chemistry. And Richard couldn't think of something easy for her, so he referred her to me, and I said, hey, why don't you make a radio-labeled ketone body? So Stephanie is working on that now, and she's at University of Alberta with Dr. Ralph Schermacher, who's a world authority on fluorine 18 synthesis. So to be able to make a radio-labeled ketone body with fluorine 18 could be something that's very important, because it might be able to trace the behavior of cancers in a new way. And that's something I'm looking forward to very much. She's making progress, and I'm really excited about that. Excellent. No, all is a good word to avoid. Many cancers do, possibly most, are glycolytically dependent. A couple of notorious exceptions are most prostate cancers, for example, are not glucose dependent, and it's a common cancer. About 80% of prostate cancer is a very slow growing and indolent, and they're not likely to take up much glucose at all. In fact, they're not approved for Medicare reimbursement because most of them don't take up fluorodeoxyglucose. About 20% of prostate cancers can be very aggressive, and those I don't know, because since they're not reimbursed, we don't usually study them. They might take up FDG. However, the aggressive ones also have a tendency to switch to glutamine metabolism pretty quickly. So they might not also. I don't know the answer. As far as other cancers are concerned, the rates of FDG uptake are pretty high. I mean, between 50 and 80% in most of the other cancers, but not universal. So it's, but it's still quite prevalent in most other cancers. Yeah, that I have almost no doubt about. I mean, that's really kind of an alarming and probably not justified statement on the basis of what I've done, because the 10-patient study was set up to be a safety and feasibility trial only. I mean, you always try to see if you have any efficacy, but it was designed as that. And on the basis of 10 patients, I can't make a generalized statement. However, truth be told, it's a very safe diet for virtually everybody, and so it's a little hard for me to believe that it wouldn't be safe. Now, I have a caveat. I mean, I did try to avoid most patients who were too thin, because this is thought of as a weight loss diet, and I guess I would say that maybe ketogenic diets still shouldn't yet be given in patients who have cachexia and anorexia, who are too thin and wasting in end-stage disease. It may turn out that that's exactly untrue, and that in fact it benefits them the most. I don't know, but for now that may be a group that I'd avoid. Okay. Yes, they do. the insulin receptor expresses itself on most cancers, and so high levels of insulin upregulate pathways that result in increased growth and tumor proliferation and resistance to cell death, apoptosis. So insulin is a signal for cancer growth, no question about it.
So inhibiting insulin is going to downregulate those pathways, I assume.
In many cases. Now, one thing I didn't mention in the talk, because it sort of took me to the 45-minute mark and I didn't have time to go into more detail, but many cancers have mutations in these pathways that upregulates them what they call constitutively, which means independent of insulin. And one of those pathways, I mean, I think I mentioned in the talk, they sound like alphabet soup. You know, there's the mTOR pathway, and then there's the RAS, RAF, MEK, ERK pathway. So these pathways are downstream of the insulin receptor, and not uncommonly, these are mutated to be constitutively activated, in which case insulin may not have as beneficial, reducing insulin may not have as beneficial effect in cancers that have those mutations. But the beauty of a low-carb diet, as I see it, is that it has multiple prongs of attack. One is through the insulin pathway and its inhibition, and the other is through, if you go on a ketogenic diet, you're also producing ketone bodies, and ketone bodies independently have an effect on cancers. At least in cell culture, we showed in our study that I did talk about that ketone bodies can inhibit cancers, can inhibit cancer growth, and others have shown that ketone bodies have histone deacetylase activity, which inhibitor activity, I should say, which others have shown can also inhibit cancer growth and proliferation, and also cause apoptosis, in other words, tissue death. So I think there are a number of different avenues by which very low-carb diets still might help cancers, even independent of insulin. Yeah, it goes back to Randall and Newsom in the 60s and 70s that they did some very interesting studies where, under a condition of an insulin and a glucose clamp, which means you maintain insulin at a constant level and glucose at a constant level, that under those circumstances, if you feed these animals, and this was done in animals, glucose, or you infuse glucose, if you infuse fatty acids or ketone bodies into these animals, that glycolysis will be inhibited. That's what it is. That's what they showed. Glycolysis will be inhibited, and it's probably inhibited because of inhibition of a couple of enzymes along the glycolytic pathway that I mentioned yesterday. Pyruvate dehydrogenase is inhibited by acetyl-CoA, which is in fact the first product that either ketone bodies or fatty acids end up making that would enter the Krebs cycle. And the second is a Krebs cycle intermediate citric acid, citrate, which goes back and inhibits pyruvate phosphofructide kinase, one of the enzymes in the glycolytic pathway. So both fatty acids and ketone bodies have the potential to inhibit glycolysis under conditions of constant glucose uptake. That doesn't usually happen in a free-living individual in the modern world if they're eating a piece of chocolate cake, because insulin will dominate under those circumstances. But if you have that clamp-like situation, which you do have in cancers, because they express GLUT1 transport, and GLUT1 transport basically guarantees a constant influx of glucose. And that's the clamp situation. And also, if you can clamp the insulin by having the patient on a very low carbohydrate diet. So therefore, you basically have an insulin and glucose clamp, but in a patient who's on a very low carb diet and giving them, and for the cancer cells. So it would reproduce the circumstances of a Randall cycle. Okay. Inhibition of glycolysis. Yeah, so I think we've been discussing this around this already. I think that if it's going to work, it's going to be a combination of both, but it's also going to depend on the cancer. If the cancer has mutations that upregulate some of the signaling proteins that constitutively activate growth, and then it may be that it may operate only through ketones. So it may have variable effects on different cancers, depending.
Okay.
And it may not work in some cancers. That's something I have to, I have to be willing to acknowledge. But I don't think that's a harm. I mean, the point is that if this can help 30% of people with cancers, or 50%, or 80%, it's still helping people, and it conceivably can help reduce the doses of chemotherapy that they may otherwise get.
Right.
And therefore the toxicities.
Mm-hmm.
In the presence of high glucose, that I don't know. We didn't really test that. Well, I had some data that I didn't present in the talk, so that this is actually pertinent. I mentioned that, I think in the talk, that 4 of the patients who were the 4 heaviest, and 3 of whom had fasting blood glucoses of 100 or more, basically had evidence of prediabetes. And we excluded patients with frank diabetes, which is to say diabetics who were on medications. Basically, we didn't want to treat two diseases at the same time. It would just get complicated, and I didn't feel that that was an appropriate way to go ahead. But if you could, but we didn't screen for prediabetes, and we weren't even thinking about it. We just thought as long as a patient isn't on diabetic meds, we'll go ahead. And, but it turns out that the patients that had the evidence for prediabetes were the 4 who actually didn't respond. Now, their blood glucoses were not very high, and we did measure their insulins, although it was a little harder to correlate that. It's possible that they had some evidence of insulin resistance, and I don't know. I mean, I just don't know whether that was the reason, but at this point it would be something for further study to determine whether or not patients with frank diabetes and elevated blood glucose would therefore be somewhat more difficult to treat, at least in the initial term. These were the ones who progressed. So, I mean, it seems strange because on the one hand you think, well, you want to give a low-carb ketogenic diet to treat diabetes. I mean, there's really good evidence that that's a terrific thing to do. But maybe it wouldn't be so good if they also have cancer. I mean, I don't know. I don't know how that balance would turn out. You would be treating 2 diseases at the same time, and now I'm not sure. I don't know what the outcomes would be. That definitely requires a lot of thought and a lot of study, and I haven't done it.
Interesting.
Ketone supplements, however, don't replace the insulin-inhibiting effects. So a diet kind of gives you all at once, and of course, depending on the tumor, if they have mutations downstream of the insulin receptor, then maybe you don't need to go on a diet. Maybe a ketone supplement would be the only thing that would be working anyway, and maybe that would then be a better approach. What was the other part that you mentioned? You mean drugs?
Yeah, anti-glycolytic drugs in combination with a ketone supplement.
I see. Yeah, well, again, that's not going to replace the diet either, but again, I think that's just going to require further study. I mean, drugs like 2-deoxyglucose, for example, I know that Colin Champ I think it's an interesting and excellent idea. We'll have to see how the studies turn out. I'm reluctant to make predictions. Yeah.
Right.
Yeah, not that I'm aware of. Acetone, which technically is a ketone body, I guess, dissipates too quickly to have any biological use at all. It spontaneously gets either destroyed or exhaled. And the other 2, acetoacetate and beta-hydroxybutyrate, I think they act pretty much interchangeably. Acetoacetate is more unstable, and it tends to convert into beta-hydroxybutyrate largely anyway. So the principal ketone body in the body is beta-hydroxybutyrate. We used acetoacetate in our cell culture studies mainly because we wanted to see whether this worked in the presence of glucose. So in somewhat an answer to your earlier question, what about in the presence of high glucose? We did actually have 10 millimolar glucose in our cell culture studies, and we also had 10 millimolar beta-hydroxybutyrate. We tried that at first, and we also tried 10 millimolar acetoacetate. The beta-hydroxybutyrate gave erratic results, and so we went to acetoacetate. Now, the reason we believe that that's true is because the glucose actually raises NADH in the cell, and beta-hydroxybutyrate has to get past a reduction barrier. In order to get from beta-hydroxybutyrate to convert to acetoacetate, it needs NAD, not NADH. If the NADH is too high, it pushes it in the wrong direction. So it was our speculation that that's the reason in cell culture that we couldn't get the beta-hydroxy to work. But in the body, I think they're interchangeable. Okay. And I don't think acetone has a role. Yeah, okay, I think we did address that a little bit before. That basically goes, that speaks to a couple of mechanisms that both of which actually have been shown only in vitro. One of which is that I think that the ketone bodies through the Randall cycle inhibits glycolysis, and also because of the overexpression of uncoupling protein 2 in many, if not most, cancer cells, that ketone bodies also they inhibit glycolysis, but they don't make compensatory ATP in the cancer cells. And that has two effects. One is that insufficient ATP is necessary for cell growth and proliferation, and we saw that. But second, actually, the inhibition of glycolysis means, and others have pointed this out, I should have mentioned it as well, is that through glycolysis you also go to the pentose phosphate shunt, which builds other anabolic molecules that are important for cell growth. So by inhibiting glycolysis, you're inhibiting cancers in a number of ways. And you do that through ketone bodies, which also don't make compensatory ATP. So you have a number of things going on at the same time. All I can say is, given the ambiguous state of my my religious affiliations, God bless Craig Thompson. No, I met Craig Thompson and Lou Cantley, both, who are really international experts on metabolic cancer therapies, at a meeting a couple of years ago, and was privileged to sit on a panel with both of them. And they both are on low-carb diets, which I found out actually through Gary Taubes. So I guess he pries a little bit. But the interesting thing about that is I've spoken at Sloan Kettering on a number of occasions, and because he's president of Sloan Kettering, I thought he might have changed the culture there. But I guess it's hard to change a culture even when you're the president of the hospital and you're Craig Thompson. So I know that I agree with him, and I think that that this is it's an exceptionally important point to make. But I've had comments when invited by the endocrine group to speak at Sloan Kettering on one occasion. They put flyers out, they put posters out, they contacted the oncology group, and not a single oncologist showed up at my talk. And so, that was a few years ago. I spoke again actually last September, and again, not a single oncologist spoke. So, my suspicion is that when you become president of a university, you just become incredibly busy. And he may not have the opportunity really to, he can't twist people's arms in that way. I mean, they do what they do, and they're getting grants, and so he's happy, I suppose. But he may he's expressing his point of view, which I think is, a valid one. Right. But it's fascinating how hard it is to get things done.
Yeah.
I haven't usually framed this in terms of macronutrient ratios like 3-to-1, 4-to-1, and I haven't even calculated it. Somehow I didn't grow up with that. I just grew up with the carbohydrate number, and the protein really was, the reason protein was kind of ignored for this study was that we were recruiting patients who were adults and who had been on generally other diets their whole life, and trying to get them to suddenly change to a ketogenic diet had to be as simple as possible. I wasn't going to do glycemic index, which I don't think you can explain to anybody, much less an average person. You know, well, I want you to eat these glycemic index foods. What does that mean? On the other hand, you can explain carbohydrate restriction very simply, which is one of the reasons I think it's a much preferred approach. Avoid all white foods except cauliflower. I mean, that's basically it. You know, you don't eat bread, pasta, rice, potatoes, and you're okay. So it was much easier to describe, but it still wasn't going to be easy to implement, and this is with a dietitian coming in and haranguing them. So I didn't really want to get into the problem of how much protein you should eat also, aside from the fact that protein recommendations, you know, are as bad as carbohydrate recommendations when it comes to the politics of it. You know, some people recommend 20 grams a day and others recommend 300. So where am I going to come in? How much am I going to tell them to eat? Ratio of protein, you know, and carbohydrate, you know. So I just wanted to keep it simple. I wasn't going to change their, you know, what they like to eat any other way other than stay away from these foods. So that's why I dealt with it that way. So I don't really have a good handle on that. I think that, you know, when I looked at the actual data, I didn't report on this in the talk either. The protein intake of the patients was variable. You know, we did get nutrition recall, and the protein intake was somewhere between usually 60 or 70 grams, and some people up to about 120, 130 in some others. If I had my druthers, I probably would want a little less protein intake on average. Maybe 50 to 70 might be enough. Maybe 50 would be enough. It wouldn't be enough if you're a performance athlete and you're trying to rebuild protein, but it might be enough for a cancer patient, and it might keep the glucose levels even lower and the ketogenesis higher. So that's just my general sense of it.
So how often are you checking their ketones, or is that one way of making sure they're in compliance or knowing they're in compliance?
Yeah, checking ketones was essential. That was actually the only way to definitively check whether they were in compliance, because nutrition recall is pretty inaccurate sometimes, and you just don't really know, you know, irrespective of what they say they ate, you don't really know. But yeah, ketosis was going to be the major measure of compliance and metabolic responsiveness. And we were measuring it in this trial just once a week, fasting bloods and fasting ketones. So that was how we did it. In this new trial, if we can get ketone bodies more frequently, that would be helpful. That's a great question. The idea of when to do a ketogenic diet with respect to other therapies, I think, is also not clear. It may be hard to do a ketogenic diet while you're on chemotherapy. The idea of calorie restriction, for example, that Walter Longo advocated either before or after chemotherapy to minimize its side effects, was interesting. So I don't really know. The drug that I chose for pursuing going forward for my own research, which I'm hoping also to do, which is to couple a ketogenic diet with rapamycin therapy, is that rapamycin-specific side effects include hyperglycemia. And so if you do that together with a ketogenic diet, you're minimizing the side effects, potentially, of rapamycin. And you might also reduce the dose of rapamycin, which would minimize some of its other side effects, which include some immune deficiency issues. So if you could do that together, that might work actually very well. Chemotherapy is a little different. It makes you sick while you're taking it. Rapamycin doesn't make you sick in those ways. It's not so much a nausea, you know, kind of an issue. It's not stopping cells from dividing and causing your colonic cells to stop dividing and giving you all those side effects. So it might work at the same time as rapamycin. Now, you know, long term, would that work? I don't know, but since a chemotherapy course or even maybe a drug, you know, a signaling modulator course such as rapamycin might be a matter of several weeks, maybe 4 to 6 weeks of therapy at a time, then you could give the diet coincidentally with that kind of a drug. Again, with other drugs, I don't know, and I'm not sure how that would practically work out. So that's what I'm really envisioning for the for using it with a drug like rapamycin, which I'm going to have to pursue that, but I haven't been as active in pursuing that yet. I want to sort of lock in this one right now because I have to get this to the protocol review committee and the IRB.
Right.
Yeah.
So could inhibiting the mTOR, could that contribute to cachexia?
I don't think so. I think that inhibiting mTOR is, insulin inhibition, which would inhibit mTOR if it's working, isn't a problem in normal adults. I think that actually cachexia is poorly understood, but is usually a result of the cancer and something that the cancer is doing. If we're successfully treating the cancer, I think that we will reduce cachexia. Anorexia, poor appetite, could be a result of some toxic chemos, like some of the heavy-duty chemos. I think that if that's not been reported with rapamycin. So I'm trying to target this in a way that might work, at least according to what I'm thinking about. Yeah. That's another great question. For the study that I'm looking at now with the 65 patients, they're actually not going to be getting PET scans because these are patients who are just diagnosed with breast cancer, and they're actually going to be monitored. The tissue markers in the tumor are going to be monitored at biopsy and at surgical removal of the tumor. And during that period, they haven't gotten a PET scan. They're just actually being worked up for determining what their therapy is going to be. And during that period, 45 of them will be on the ketogenic diet and 20 will be controls. So all we're really doing, this is a biomarker study and a change in biomarkers to see whether the ketogenic diet favorably influences markers of growth, proliferation, and apoptosis at cell death as compared to the standard diet that the other 20 patients will be on. So this particular study will not have a PET scan. Going forward, again, I don't know yet. I have to think about some of these things carefully. I think that a PET scan is a very useful way of monitoring these patients. RECIST criteria, there are PERSIST criteria and other ones that have now been established for PET scans. I guess that would be a way to go. I haven't thought about it yet. I haven't been very happy with RECIST criteria to begin with. I mean, everyone uses them, but I think they're terrible. The RECIST for CT and X-ray findings use one dimension of a tumor that's three-dimensional and you could be missing everything. And the only reason it hasn't changed is because it's hard to change a standard when it's been accepted for the last 15 years. Someone has to do a very expensive study to prove that a new standard is better than the old one. So I don't know what standard I'm going to use. But anyway, but I do think that the PET scan is going to be in there. It's going to have to be in there.
What are the biomarkers that you're going to follow?
Well, the two clear biomarkers we're going to follow, one of them is called Ki-67 because it's a marker of tissue proliferation. And the other marker we're going to follow is tunnel, because tunnel is a marker of tissue death, apoptosis. And we want to particularly see if those two are influenced. If we can also get a liquid biopsy, which is to say that there are now very sensitive assays where you could actually measure tumor cells in the blood that apparently are being shed, and this is a very early marker for potentials for metastasis. This is something that we would like to see too. I don't have an hypothesis yet as to why in 3 weeks we might change a liquid biopsy. So, but if it's available, I would just be interested to see if it does change. So there are apparently these newer techniques, and we would like to include that one if we could get it. Actually, I've chosen to look at breast cancer because Joe Sperano, my biggest supporter, is a breast cancer oncologist. That's the principal reason. The original study, as I said, was chosen. He was game for looking at this in general, and I was expecting that because we had a weight limitation, we wanted patients not to be underweight, and yet we wanted them to be advanced. It happens that breast cancer and colon cancer are two cancers that can be rather advanced and still have patients that have not lost much weight. So I was expecting that the majority of our patients would be breast or colon. And But it turns out we also ended up surprisingly getting a couple of patients with lung cancer, a patient with esophageal cancer, 2 with ovarian cancer, so I didn't expect that. So it turns out we got a greater diversity of cancers, which has some advantages and has some statistical limitations because there are very few. But the real reason for breast cancer alone right now is that I know I have a source of patients that I'm going to get referred to. But I'd be willing to look at any cancer, frankly. I think that this is a widespread use for a variety of cancers, and glioma is one of them, but it's not the only one.